Hello, everyone. Welcome back to the Genomics Bootcamp and to a presentation about PLINK genotype input data formats. I wanted to do this video for a longest time, and that is because it is really useful to know what are the input data formats and exactly what structure they require. And the reason why it is very useful to know all this about these data formats is that sometimes you have your data that you eventually want to analyze by PLINK or with PLINK is in a, well, just a strange format. So basically knowing all the possible data input formats, this might cut down the time to modify your data so you can get it into PLINK. So by knowing about all the possibilities of input, you might find out that your data is actually quite close to some of the formats. And then you can cut back on the data preparation time. And whoever did data preparation or data modification before can confirm you that you want to minimize this step as much as possible because it is a pain most of the time. So anyway, I will present you the PLINK input data formats. Of course, you can find much more information about these on the PLINK website. And also I will try to put some links into the description of this video. The first thing to mention is the very well-known binary genotype format. This has, well, 3 files that it requires, and these three files have the same, well, the first part of the name is the same. Well, in this particular example, I always will name them or call them my data, but basically with the extensions .bed, .bim, and .fum. So these 3 files should be present in your directory so you have the complete information about your genomic data. We will go into the details of these files in a minute. If you want to work with the binary genotype format in PLINK, you have to use the --b file option. So the FAM file is basically a small text file that contains information about all the individuals. It has, well, several columns, and the first column is the family ID, then the individual ID, then there is the ID of the male parent, female parent, sex of the individual, and phenotype. Well, there could be occasions that these are known and then they are properly filled in here. But in my particular example that I'm using, this was just not known, so I put them as unknown with a 0 or a -9 in case of a phenotype. By the way, for this demonstration, I will be using the same data all the time, and that is some Hereford cattle data from open access resources. But actually, it doesn't matter from which species the data is. I just wanted to show you also not just the, well, the column name, but also how the file looks like, or more or less what columns could be expected. So anyway, this is the .fam file. And then the next one that is also required for the binary genotype format is the .bim file. It has also, well, 6 columns. The first one is the chromosome, then the variant ID. So this is basically the SNP name. Then the position in Morgans and centimorgans. Well, this could be a 0. Of course, if it's known, it's fine. If it's not known, it could be a column of zeros. Then there is the base pair coordinate. So this is basically the SNP position, and then allele 1 and allele 2. So in this file, each line is a SNP, and basically it gives you the map position, so the SNP coordinates. Well, the third file that is required are actually the genotypes, but these are in a binary format, as also the name suggests. So these are not readable by human eye or with a text editor. And if you open it, you get this kind of gibberish. But the computer recognizes these binary files very well and can work with them in a very efficient and fast manner. So as far as efficiency and speed is concerned, I believe these binary genotype formats are the best solution for your datasets. But of course, there are many others that could be useful in many other occasions. So this was the binary format. And now I move to the PED and MAP file, which I call here just as the classic, because for me, at least, these two files are the most iconic one when I think about PLINK. So, well, classic maybe just for me, but certainly one of the most used and most popular data formats as for the SNP data. Here, the expected file names are, or actually file extensions are the .ped and .map. And if you want to read them in PLINK, you have to use the -- file option. So the PED file has a very specific structure. The first 6 columns are basically the FAM file. So this 1, 2, 3, 4, 5, 6 up to here to -9. So this is basically the FAM file. And basically it has the same columns as I mentioned earlier. So I'm not going to repeat those. But what follows actually are 2 columns for each SNP. So basically in this file, in the PET file, each line here is an individual, and then the genotype of the first SNP are these 2 alleles. So the first SNP is a homozygous AA. The second SNP is a heterozygote AG. The third SNP in animal 1 is also a homozygous AA, and so on. So then it goes into this right side here. So this file is actually very wide, and depending on how many SNPs you have, so there are twice as many columns. A very useful feature of the PET files is that they contain the genotypes, and they could be also opened with the text editor. So if you want to check the exact genotypes, well, PET file is certainly an option. The map files that are also required together with the PET file are quite similar to the BEAM file as we've seen before, but without the allele codes. Well, it has 4 columns. So first one is the chromosome, then the variant IDs, which is basically the SNP name. Again, Morgans or centimorgans, which could be 0 or, well, a number if it's known. And then the fourth one is the base pair coordinate, or the base pair position for each SNP. The next input data format is the so-called transposed PET file. Here we have also 2 files that are required. So the first one is, well, with the .tped extension, and the second one is with the .tfam extension. If you want to read these in PLINK, you then need the -- file option. Now, this transposed PET file could be a bit confusing if there are many animals, so I limited for this particular example the data set just to 3 animals to be able to show you how it works. Or, well, of course, we could do it also with more animals, but this, I think, it's a clearer example. So anyway, what we see here, each line in this tped file is a SNP. So you have as many lines in a tped file as many SNPs you have. The 4 columns, the first 4 columns of the tped file are basically the map file. So you have the chromosome, you have the SNP name, the Morgan, and the SNP position. And then each 2 columns after that are the genotype for the respective animals or individuals. So basically, the first individual has the genotype AA is actually, all of them have AA here. But well, let's see, for example, this one. That is, well, the first animal has a GG, and second one is AG, and the third one is AG as well for this particular SNP. So it's basically similar as the PET file, but in a transposed way, as also the name indicates. So while in the PET file, there were animals in lines and SNPs in columns, in the tped file, there are SNPs in lines and animals in columns. I mentioned that there is also a second file, which is with the tfam extension, but in reality, this is exactly the same as the FAM file. So it has just to have a different name, but otherwise it's exactly the same. The next format that you could encounter also in the videos on the Genomics Bootcamp is the so-called long format. For this data input, we require 3 files with the specific extensions with the .lgen, .fam, and .map. And if you want to read it in or use this in PLINK, you need to use the --l file option. I mentioned that we already seen these files on the Genomics Bootcamp for sure, because if you remember, if you want to transform data from the so-called final reports to PLINK files, transforming them into LGen files is a very convenient middle step. So how these LGen files look like? Well, in here, each line is one SNP for one individual. So if you have, 50,000 SNPs, then you will have 50,000 lines for individual 1, 50,000 lines for individual 2, and so on. It has a very specific structure of columns. So first one is the family ID. The second one is the individual ID. In PLINK website, also often referred to as the within family ID. So that is, this one. Then there is the variant identifier. So that is basically the SNP name. And then allele 1 and allele 2. Also with the LGM files, that there is a new thing that also you can actually consider or set the reference allele as well. So if your work requires this, well, it is certainly a possibility. I mentioned that also in the long file format, the .fam and .map files are required, but these in fact are exactly the same as shown before. So the FAM file with the individuals and the .map file with the SNP coordinates is exactly the same as it was shown in the previous slides. The next option, which is also very prominent and very convenient, especially if you work with the whole genome sequence data, is the so-called variant call format or the .vcf files. Indeed, the VCF files could be directly loaded into PLINK. Also, there is an option for BCF files, so which are the binary versions of VCF files, so these could be also loaded. If you want to do so, you should use the --VCF or --BCF option. As I mentioned, the VCF files are the standard output files from whole genome sequence analysis, and therefore you might come across them quite often. Here, I also want to warn you that, well, here is also a possibility or even a necessity to consider the reference alleles. There are a lot of additional options and possibilities to do so in PLINK. So if you are loading or dealing with VCF files, I definitely recommend to check out these in detail. Also, the VCF files are one of the more complicated file formats, and I don't want to go into very much details on the VCF format. Maybe we do another video some other time, but for this particular purpose, I just show you the most important part, which is the header information and some of the columns. Here also each variant is a line or a row in this particular file. You might also notice that, well, because of the text editor, some of the column names are actually a bit shifted, but basically this is the chromosome, the position, the SNP ID, reference allele, alternative allele, and so on. After some time, there is a column for each individual. So this would be for individual 1, individual 2, and then also the other individuals here, which indicate the genotype. Also, I want to mention that the VCF files are also very good, not just to showing SNPs, but also you can indicate insertions and deletions and other variants in the VCF file. So in terms of scope, it has much larger possibilities. The next input option to read in the genotypes in PLINK is the so-called Oxford format. Here, there are two files expected, so the .gen files and .sample files. Also, well, there is a binary version, so the .bgen is basically the binary version of the gen files. If you want to read in the genotype files into PLINK in the Oxford format, you need to use the --data option. So first, let's talk about the .gen file. Here, each line is a SNP, and similarly to the bin files, there are the first 5 columns indicating the SNP positions. Basically the same as the bin file, just without the Morgans. So this is what I mean. So the first one is the chromosome, which is, well, indicated in a PLINK in a, well, bit of a strange way, because there are, well, other possibilities, etc. But also, this is the reason why is the SNP ID is in the quotation marks here. So this is basically the chromosome. The second one is the RSID or the variant ID or the PLINK name, basically. The third one is the base pair coordinate. And then it is followed up by allele 1 and allele 2. But then what is very interesting is these 5 columns are followed by 3 columns for each individual, indicating the likelihood of a genotype for each sample. So first, I show you what I mean. So this is then the, well, basically the bin file or the first 5 columns. So the chromosome, SNP name, position, allele 1, allele 2. And then, well, these 3 columns are for individual 1, and that's indicating that it's a bit, so this is homozygote, heterozygote, homozygote. So this indicates that the first individual is a heterozygous one. The next 3 columns here are for the second individual. So this is in this is 0 likelihood for homozygous, 0 for heterozygous, and 1 for other homozygous. So this indicates that it is homozygous for a G allele. And the same way, the genotypes are coded in this format. Here we have, well, zeros and ones because we are talking about SNP data. But as I understood, these values actually could be different from 1, or, well, basically just indicating the likelihood in some decimal numbers, which then opens up a whole range of other possibilities. Well, the first file for this Oxford format was this .gen file, and as I mentioned, the second file is the .sample file. Well, it has also a bit of a more complex structure, and in this case, I actually copy-pasted the part from the PLINK website to show you. So basically, it has 2 header lines, so header line 1 and header line 2, and then these are the contents. So basically, it comes down to a similar situation as before. So we have the family ID, the within family ID. So basically, this is the individual ID, the missing allele frequencies, 6 codes, and phenotypes. So in a way, this is somewhat similar to the FAM files, except there are no parental information, but there is this one more column with the missing call frequencies. And the next option, what you can use in PLINK is the so-called 23andMe file format. Well, this is quite restrictive because it can be used, or, well, if you try to use any other species, the PLINK will give you a message that it can be used only for humans, and also it can contain only 1 person. So each 23andMe file format contains 1 human genotype. There is no specific, well, file extension. So the .txt files are fine. And if you want to load them in PLINK, you have to use the --23 file option. So what columns you need to have in this file format? Well, this is quite fixed and given. So here each line is a snip, contains, well, the header part or with information, RSID or SNP ID, chromosome position, and genotype. It looks more or less like this. So there is this information and pretty straightforward RSID, or, well, basically the SNP ID. In this case, well, I was also modifying the cattle data to trick the system a little bit. So I just can show you some example here, but normally these would be human SNPs, or because this would be a human data set. But anyway, so these are the SNP names, chromosomes, positions, and the genotypes, which are shown here in this joint format, so without space between the alleles. Well, this was the last thing. I tried to look up all the possibilities for data input or genotype data input in PLINK. I hope I didn't miss anything. If by chance something is missing, please let me know in the comments below. And because you remained until the very end of the video, I give you a little teaser for one of the future videos on the channel. That will be the genotype output file formats, which can be done with PLINK, which are also very, very useful to know. For today, I thank you for your time and have a really nice day.